Hello, everyone, and welcome to today's AAPS eChalk Talk, hosted by Sapient. You can submit questions at any time by going to the questions box of the GoToWebinar platform. All questions will be seen by today's moderator, and we will be answering them at the end of the program. It is my pleasure to introduce our moderator, Tanya Nguyen. Tanya, the floor is yours.
Thank you, Stacy, and hi, everyone. My name is Tanya Nguyen, and I'm Head of Scientific Operations here at Sapient. I'm very excited to welcome you to today's talk, in which we'll be discussing the clinical trial conundrum, identifying responders versus non-responders. Identifying patients who are likely to respond to therapy is a key challenge for any clinical trial. In this presentation, we will explore how small molecule biomarkers can be discovered and applied to improve patient selection and enrich for responders. I think you'll find this topic quite timely and hope it gives you some valuable takeaways. To kick things off, I'd like to introduce our speaker, Dr. Mo Jain, who is CEO and co-founder of Sapient. Dr. Jain is a physician scientist with more than 20 years of expertise in physiology, computational biology, and mass spectrometry-based metabolomics. Prior to founding Sapient, he formed and was director of the Jain Laboratory at the University of California, San Diego, where he led his team to develop next-generation mass spectrometry systems, what we now call rapid LC-MS, to probe the non-genetic landscape of disease across large-scale human studies. Dr. Jain found Sapient in 2021 as a spin-out of the Jain Laboratory to expand upon this mission. Mo, I'm looking forward to hearing your take on the clinical trial conundrum.
Oh, thank you very much, Tanya. And we're excited to be here today, and I appreciate everyone who's joining us online. And as Tanya mentioned, we really want to spend some time today talking through this fundamental conundrum that everyone in biomedicine is really faced with, and that's to respond or not to respond. How do we begin to identify those individuals who benefit most from any given therapeutic, and how do we utilize this information to accelerate the drug development process? And we're hoping over the course of the next half an hour or so to present to you some ideas that we have from Sapient, as well as present to you our approaches for solving this underlying conundrum. To begin with, and just to frame out the problem, and certainly anyone who works in drug development is well aware of this, typically the drug development cycle starts with thousands of compounds that are being screened to really target a fundamental biological process. And even after a drug enters into clinical trials and clinical studies, it's well known that only 1 out of 10 drugs will ultimately emerge to gain regulatory approval and be used clinically. When we think about this and begin to break it down by actual phases of clinical trials, what we find is that among those agents that enter into clinical studies, 70% of them fail at the phase 2 level. Now, arguably, this could be expected as phase 2 is really meant to serve as a proof of concept and to provide some early evidence for the underlying efficacy. What's more surprising, however, is that over 50% of clinical trials also fail at the phase 3 phase. Now, these really should serve as large confirmatory trials that theoretically have a lower failure rate, but as you can see here, more than half the studies that are entering into this ultimate stage continue to ultimately fail. Now, we've spent quite a bit of time at Sapient and in the scientific literature really dissecting down this question of clinical trial failure and understanding why clinical trial failures occur at this very late stage when time is of the essence, when patient lives are at stake, and when obviously expenses are quite great. And what we found is that 57% of phase 3 failures are due to actual lack of clinical efficacy. Now, when we break this down and really dissect as to why this may occur, this broadly enters into 3 ultimate categories. The first has to do with how we begin to think about human models, the lack of translation from animal models or lack of translation from phase 1, phase 2 studies to ultimately confirmatory phase 3 studies, insufficient measures and understanding of the underlying pharmacokinetics and pharmacodynamics also contributes to this early discrepancy in what we call human studies. The last two bins here, the one shown in the middle and the one shown to the right, really frames out the majority, however, of these phase 3 failures. When we think about the lack of efficacy, this is oftentimes a lack of statistical significance. And this can be broken down into several different areas. One, clinical trials are often underpowered. The inclusion criteria are oftentimes poor or too broadly defined to ultimately allow for statistical significance in a subset of individuals. And obviously, many of us have been faced with the high rate of placebo response, which can convolute ultimate statistics. When we think about actual enrollment and the heterogeneous nature of both individuals as well as their disease processes, it's clear that there's variable disease response as well as variable host response to a particular drug that also greatly influences ultimate efficacy at a phase 3 stage. Now, these latter two bins, the lack of statistical significance and the underlying heterogeneity that we see within both disease as well as individual populations, really point to structural issues with how we frame out phase 3 trials. And the concern is that many drugs that are truly efficacious may be artificially failing due to these underlying structural issues. And we're hoping to talk through now how we can think about some of these, what may be efficacious drugs, and how we can use biomarkers to rescue or to better advance these agents. Now, what's particularly surprising is that even if we look among those FDA-approved drugs, and here are 30 or so clinical studies for which ultimately resulted in FDA approval based upon a surrogate endpoint, what we find is that the response rate is greatly variable. These response rates range even within the cancer therapeutic space, which ultimately uses targeting anywhere from 80% all the way down to below 20%, with a median response rate, again, for these approved FDA agents of approximately 41%. What we're finding is that across multiple clinical studies for the same agent, that the more heterogeneous the underlying disease, the more the response rate drops, and ultimately this necessitates larger study sizes that includes enough responders. Now, this is not something that's lost on the underlying regulatory agencies, including the FDA. And as many of you are aware, the FDA has actually provided what they call biomarker-guided enrichment strategies to enable the selection of patients as a way of entering individuals and allowing for enrichment among responders in clinical studies. Now, these types of markers can broadly be brought down into three areas. The first is markers that identify a specific aspect of pathobiology or pathophysiology. An example of this would be a biomarker that characterizes patients with a fast progressing versus a slower, more indolent progression of the disease, in which a drug would likely have a beneficial effect under a shorter period of time. The second type of markers are those markers that indicate a past history of response. For example, a biomarker that identifies patients with early clinical response in an open label pre-randomized period of the study. And finally, the third type of markers are those that characterize the fundamental disease or the drug's mechanisms of action and aligns that with a particular disease state. And the classic example of this from the oncology literature is a biomarker of an overexpressed gene, which the drug is targeting and enriching patients for these individuals. Now, there's quite a bit of evidence from the clinical literature that is, in fact, this is not a theoretical benefit, but an actual benefit. This is an analysis of now over 20,000 clinical trials from Andy Lowe's group at MIT back in 2019, when they studied those factors that allowed for approval or successful completion of phase 3 clinical trials versus those that did not. And among all the various factors studied in this underlying paper, the one factor that was universally associated with increased drug approval was the use of biomarkers. This was irrespective of the therapeutic area, whether it be in the oncology space, whether it be in the cardiovascular space, autoimmune disease space, infectious disease, vaccine development, et cetera, et cetera. And when we look at over 20,000 trials from the last several decades, we find that there's over twofold increase in approval rate when a drug is developed together with an underlying biomarker. Not only is the approval rate higher, but the time to approval is much faster, with an over 20% improvement in the actual time it takes to approval for those medications that are developed under what we call a precision-guided mechanism, whereby a biomarker is actually used to guide the implementation of the underlying drug. Now, here's one such example, a classic example from the literature for the phase 3 agent trastuzumab that had been initially tested in breast cancer. And what we found, as you can see from the table here, in a non-enriched trial, which included multiple individuals with breast cancer, over 8,000 individuals, the response rate was 67%, and the underlying p-value was found to be non-significant. Now, in subsequent studies, it was identified that trastuzumab is working particularly through antagonism of the HER2 new biomarker and the HER2 new receptor. And when we use that receptor as an actual marker to enrich individuals in a clinical trial, what you can find in a subsequent phase 3 clinical trial now, which we label here enriched trial, the number of individuals enrolled was 17-fold lower. So from 8,000 to under 500 individuals, the response rate increased from 67% to 78%, essentially through the targeting of individuals in which the drug would most likely benefit. And the p-value, as you can imagine now, went from a non-significant to a below 0.05 that ultimately allowed for the approval of this agent and the targeting of this drug specifically in individuals with the HER2 new biomarker. Now, this is just one such example, and there's many examples in the literature, particularly from the oncology literature, whereby taking this biomarker strategy can transform clinical trials, allow them to be completed in a much smaller subset of individuals in a much quicker time frame with much higher degree of efficacy. Now, when we think about this personalization of therapy, we oftentimes think about the host, the individual themselves. And what we find, while all of us are slightly different, the differences between us are really quite minimal, whether it be through genetic variation, lifestyle variation, etc. We really are fundamentally not that dissimilar from one another. However, what is very different from one individual to an individual is our actual disease and our disease processes. While we think of disease as a singular event, oftentimes which is defined by a pathologic finding on biopsy or study, we know diseases have many, many different etiologies that allow you to go from a normal phase to an abnormal state. And being able to classify disease according to this subsetting of their pathobiology is critical towards this personalization of therapy. Now, of course, we've seen this evolution occur in the cancer space, whereby the use of these markers to subset cancer has really proven transformative. When we think about lung cancer, going back approximately 40 years ago, our traditional view of lung cancer was really one that broke down into 3 categories, again, largely based upon underlying pathology, how it looked on a pathologic specimen. And of course, this is the classic breakdown of adenocarcinoma versus squamous cell carcinoma versus large cell carcinoma. As this advanced in the early 80s, we began to identify that there's molecular markers that can denote specific subsets of lung cancer according to particular mutation status. And that allowed us to subset lung cancers, initially into KRAS positive versus KRAS negative, subsequently to KRAS and EGFR positive in the early 2000s versus those that are known. And if we look today, there's dozens of subsets of lung cancer, each of which is defined by a specific biomarker that denotes a mechanism by which tumor genesis has occurred. And of course, for any of us in this area that works in drug development, typically therapies are targeted towards specific subsets of these individuals based upon this biomarker. Now, this has obviously worked quite well using genetics in the oncology space, particularly since these genetic measures allow us to subset tumors according to their molecular pathogenesis. But what happens when we think about the non-cancer space? Are there other markers that we can leverage for which genetics may not be ideal to identify subpopulations of disease? And here is where Sapient enters, and we spend quite a bit of time thinking about other non-genetic markers that may in fact allow us to subset disease. And when we think about these non-genetic markers, we're particularly focused on what we call small molecule biomarkers that allowed us to read out both host and disease factors that may influence drug response. Now, these small molecules represent the tens to now hundreds of thousands of small molecules that are produced within our intracellular environment that are present in our blood. Given the physical chemical properties of these small molecules, they can be produced in normal tissue, in diseased tissue, or even within the microenvironment. And given their size and chemical properties, these molecules are readily translocated from the intracellular space into the blood. What this means is we can non-invasively measure these within blood samples, and they provide a reflection of what's actually occurring at the level of the tissue across the body in a holistic view. They read out different disease pathologies. These markers can be used to read out target engagement and even pharmacodynamic response. As many of you know, these small molecule biomarkers are among the most ancient of factors in our blood, certainly evolved to a far greater degree than proteins or genetic markers. And because of that, they're highly conserved down to preclinical models and even cellular studies. Now, this is not a particularly new idea, and certainly within the literature, there's many people who have used small molecule biomarkers in order to identify key biomarkers of drug response. The challenge has always been, how do we go about measuring these small molecules at scale in a comprehensive fashion that allow us to make fundamental discoveries? Introduction of Sapient now, and this is where we have transformed the use of what we call rapid liquid chromatography, or RLC, mass spectrometry systems. These are next generation mass spectrometry technologies that now enable for the first time the comprehensive measure of small molecule biomarkers across large population scales, essentially allowing us to measure tens of thousands of things from biological samples across tens of thousands of individuals. Here's an actual snapshot image of these RLC mass spec instruments here at the Sapient site in San Diego. What these instruments allow us to do is to take any biological specimen, whether it be a blood sample or tumor sample, other biofluids such as urine, CSF, tears, vitreous eye fluid, normal tissues, microenvironment tissues, etc. And in each of these biological specimens, we can assay over 11,000 small molecule biomarkers. For each of these biomarkers, we do what's called comprehensive 4D chemical classification across a broad array of chemistries, measuring the retention time, the CCS or cross-sectional collision area, the MS1 and MS2 physical chemical properties. From these 11 to 15,000 molecules, typically we can identify 500 to 1,000 of them, assigning substructural identification to each of these molecules using this 4D chemical characterization, as well as have the means to use reductionist chemistry approaches to isolate and identify any unknown molecules of interest. Now, what makes this particularly special is we can operate these mechanisms and operate these technologies at scale with an analytical cycle time for analysis of a single sample that's now at about 53 seconds per sample. Ultimately, this allows us to handle thousands of samples within a given experiment at a given time to make robust measurements that allow us to advance forward the discovery of these small molecule biomarkers. Now, the generation of data is really only one component of this, and one lesson that we've learned over the last several years has been that data has to be integrated in a meaningful fashion in order to allow for discovery. And so while we think of this mass spectrometry small molecule biomarker data as our new data that's being generated, this data is often integrated in a very complex fashion with genomics and proteomic markers, with longitudinal clinical outcomes, with preclinical data and preclinical models, with pharmacodynamic modeling, with microbiome measures, with other metrics surrounding human biology and even clinical laboratory values. And it's really the nexus of this integration of this multi-omics approach that leverages small molecule measures together holistically with these other markers that enables the greatest degree of discovery. Now, given this factor, we've assembled tens of thousands and now hundreds of millions of data points in what we call Sapiens Human Biology Database. This is the database that includes hundreds of thousands of human biosamples from around the world across many, many different sample types, from blood to tissue to tumor. And many of these biological samples, we have decades of clinical follow-up, allowing us to examine how small molecules may map onto the development of disease over time. Now, this information, including all these small molecule measures that have been assayed in each of these biological specimens, has been introduced into a very complex relational database that includes over 100 million now phenotypic data points across a multitude of disease states, from cancer to cardiometabolic disease, maternal fetal health, autoimmune disease, neurodegenerative disease, liver, GI, and digestive illnesses, renal diseases, ophthalmic diseases, and even in rare diseases. Now, for each of these biological specimens, we have made these measurements, again, assaying over 15,000 small molecule biomarkers in each of these biological specimens, and have integrated this data with genomic measures, microbiome measures, lifestyle factors, proteomic and protein measures, longitudinal clinical outcomes, demographic factors, and response to various drugs that allow us to really enable this discovery process. To show you one just such example of it, here's how we begin to integrate genomic measures and small molecule biomarkers. So in the Sapient Human Biology database, we have over 20,000 individuals in which we have matched deep interrogation of the underlying genome, as well as from their blood measures, measures of these small molecule biomarkers. Essentially, this allows us to do genome-wide association around any single biomarker. And when we do this across ethnically diverse and genetically diverse individuals, you can see the resulting Manhattan plot, which is shown here in the bottom, whereby we can discover new genetic factors that integrate and associate with various small molecule biomarkers. This has proven to be extremely valuable as we discover these small molecule biomarkers to identify the biosynthetic pathways, the transport mechanisms, and the tissues that give rise to each of these factors in blood. So all of this is wonderful, and I'm hoping that we can take a couple minutes here just to share a couple case reports before we get to questions and walk you through how we go about making these discoveries of these small molecule biomarkers. They may interact with drug response. So the first, thinking through markers that may map onto common diseases, Sapient works across a host of different disease states, 3 to 4 dozen different diseases as we showed you in our human biology database. And in one such disease here, such as NASH, we can begin to make these thousands of measurements in large populations of individuals, essentially assaying thousands of molecules, and ask, are there new diagnostic markers that begin to identify people with certain diseases? And as shown here on this image on the left, in fact, we can identify singular biomarkers that denote people with certain disease states, in this case, a common disease of NASH. We can validate these in independent studies showing with increased stage of NASH, the median level of this marker increases. And one of the common lessons that we're continually taught, going back to the underlying reason and motivation for this talk, is that even individuals that share a common pathology, in this case, individuals that share a common diagnosis of stage 3 NASH based upon pathologic examination, can have varying degrees of biomarkers. And we see there's a subset of individuals that have very high levels and a subset of individuals that have a more modest increase in this marker. Now, this denotes individuals that likely develop this shared pathologic change of stage 3 NASH for very different mechanisms. And we have data and reason to believe that they may respond to very different therapeutics. And in a way, we subset cancers by molecular etiology. We can use these small molecule biomarkers to subset non-oncologic diseases, in this case, NASH, in a similar fashion. Now, we can also begin to evaluate treatment response and the dynamic nature of these markers using our human biology database. And as shown here on the right, when we look at individuals before and after bariatric surgery, we find these markers to be quite dynamic in change, in corresponding change with pathologic inflammation within the liver, suggesting, in fact, that these can be quite dynamic markers that can read out and be used as surrogate endpoints in early phase 1 clinical studies. We've similarly taken approaches in rare diseases, and shown here some example of a rare disease population in over 100 individuals with sequence verified mitochondrial disease. And as we know, among many of these rare disease populations, despite a singular mutation or shared mutation, there can be quite a bit of phenotypic heterogeneity, as we see in this population on the left here in this chart. Many of these individuals can have very different organ systems that are affected and very different clinical presentations, despite sharing a common mutational burden. Now, when we look among these individuals, again, measuring these thousands of molecules, we can find that there's singular small molecule biomarkers, as shown in this volcano plot in the middle, that denotes individuals with these disease. And when we look at this single marker in independent studies, what we can find is not only do they validate in individuals with systemic mitochondrial disease, but there's a massive spread, and we can begin to align the degree of this marker with certain phenotypic variables, including the degree of severity of underlying symptoms. Now, we could also begin to think about how we use these markers to uncover drug response, particularly in phase 3 clinical trials. Here's work done together with a client looking at a phase 3 clinical trial in several hundred individuals that ultimately failed for lack of efficacy, going back to our initial slides. When we analyze biosamples from this clinical trial, we identify a single marker, shown here as the red dot, that denotes individuals that actually respond versus not respond. And when we look at this single marker at baseline and following therapeutics in this population, we see there's a population of individuals that have an increase in this pharmacodynamic marker over time, whereas there's also a subset of individuals that have very flat levels of this singular marker. If we actually begin to splay out their drug response on this Kaplan-Meier plot, looking at mortality on the bottom right, according to levels of this pharmacodynamic singular marker here, what you can find is a great disparity between those individuals that actually mount a response and have high levels of the marker after therapy versus those that have low levels of the marker. Now, this is a marker that's being advanced forward as a pharmacodynamic marker in the same way we can use molecular diagnostics to identify those patients who are most likely to respond. I'm hoping to just stop there and take a couple questions, time allotting, Tanya. And if anyone is interested in following up and having conversations about this, please don't hesitate to reach out to us. You can use the QR code to schedule a meeting, our phone number, email, as well as our Calendly link is shown here. The website is www.sapient.bio, and I'd be happy to take any questions.
Thank you, Mo, for that really intriguing presentation. We actually do have a few questions that have already come in from the audience. So I want to thank those who have submitted questions and remind everyone else that you can still continue to add new questions through the chat. For now, let's start with the first question. Mo, can you talk a little bit about the specificity of these small molecule biomarkers? You mentioned that they integrate different drug response as well as host characteristics. So how can you tell if a biomarker is coming from the disease or the drug response or another part of the body?
Yeah, it's a great question, Tanya, and thank you for that. When we think about these small molecule biomarkers, as you can imagine, in a plasma sample or in a blood sample, we're integrating information that comes from a host of different organ systems. Now, there's many ways that we go about understanding underlying specificity. The first is through localization, and this is where the integration with genetics, as you mentioned, can be absolutely instrumental. By doing GWAS around any singular marker, we can oftentimes identify the biosynthetic enzymes and transport systems that give rise to that marker. And by localizing those enzymes and transport systems, we can often identify the organ system as well as the cell type, and in certain cases, the organelle even, from which the marker is emerging. With regards to underlying specificity for a disease state, this is where the human biology database is critical. As we identify a single marker that may associate with a disease or a drug response, we can look in the hundreds of thousands of individuals within our human biology database to begin to ask, what particular diseases does this marker associate, and how does that map onto various organ systems? And through that analysis, again, across hundreds of thousands of people, we can very quickly begin to understand the degree of specificity. How does this marker relate to other related diseases, or how does this marker relate, for instance, to response to other related therapeutics?
Thank you. I think that makes a lot of sense. Do you believe small molecule biomarkers are more predictive of drug response relative to traditional genomic or other protein biomarkers?
Yeah, it's a really interesting question to think about. And really this comes down in many ways to the particular disease under question. I think there are certain disease states, oncology being the classic example, which genomics has been transformative. And this speaks once again to the use of the genome to understand molecular pathogenesis of the tumor, not necessarily the host, but again, the underlying disease itself. And these genomic markers have really transformed how we think about the subsets of tumors that may respond to certain targeted therapeutics, as we showed you the example in lung cancer. Now, the challenge really comes down to non-oncology space, and for those diseases in which genomics has not necessarily been transformative. And this includes everything from heart disease to GI illness, lung disease, Alzheimer's, and neurodegenerative states, essentially all the other diseases that plague mankind. Under those circumstances, what we find is that these small molecule biomarkers can provide an immense amount of information, not only around subsetting of particular diseases according to pathobiology, but also recover responder, non-responder status. Now, this isn't to suggest that these small molecule biomarkers don't have a role to play in cancer. Certainly, I believe firmly that they do. And oftentimes, this is through an integration of both genomics and looking at mutational status together with these small molecule biomarkers can reveal quite a bit about responder status or non-responder status, safety status, et cetera, et cetera. But particularly when we open that spectrum beyond the oncology world, I firmly believe these small molecule biomarkers can be transformative in how we understand disease, how we diagnose it, how we subset it, and how we implement therapeutics.
Yeah, that's a really great point that you bring up. Moving from clinical trials to kind of more clinical aspects of how these mass spectrometry approaches can be applied. How do you think we can use this for the personalization of therapy? So how do you go from R&D discovery to an actual clinical test?
Yeah, really good question. And this is something we spend quite a bit of time thinking about here at Sapient. The goal is not fundamental science for the sake of only science, but rather, as you mentioned, rapid translation to clinical implementation. And the way we think about it is that we use these rapid screening RLCMS systems oftentimes to study large populations, to make singular discoveries through our biocomputational approaches, to validate those discoveries both biologically using our human biology database, essentially assessing the underlying validation, as well as using orthogonal mass spectrometry systems to technically validate every single measure using long-graded traditional LCMS. Now here at Sapient, we actually have a high complexity CLIA laboratory that allows us to rapidly take any one of these single markers and develop a CLIA certified assay that we thereafter can implement as part of clinical studies or clinical practice. And so we really make these discoveries with an eye towards rapid translation, which is part of our underlying mission.
Great. And I think we have one more here. In your case example, you reduced a large number of statistically significant metabolites to a single biomarker. What do you think is the benefit of doing this versus using a profile of several metabolites? How can you be sure that that single biomarker is actually a true signal?
Yeah, it's a really interesting thought, and there's a sort of a variety of answers to the question. You can imagine when you're making these tens of thousands of measurements, the goal can fundamentally be to isolate this down to a single marker that captures the majority of the information, all the way up through the use of very complex machine learning algorithms that may impute information from tens of thousands of different measurements and their interaction to come up with very complex profiles that allow for subsetting disease. Now at Sapient, we oftentimes take both approaches, and really the best approach is fundamentally related to the key question and the key goal. Implementation of clinical practice is really, as I mentioned in the prior question, one of our fundamental goals, and that practical implementation oftentimes requiring identifying a single marker or two markers or three markers, but a small subset of them that really capture the modality of the clinical information. I recognize for the examples that we showed here, I went through them rather quickly and we didn't have time to really talk through sort of the underlying statistical details, but for those cases, what we found is that a single marker captured 95% of the information, and adding on a second marker or a third marker did not provide additional information under those circumstances. Now, there's certainly other disease states and other biological questions, whether they be around drug response or diagnostics, for which having multiple measurements really is the key, and I think that firmly makes sense. On the other end of the spectrum, as I mentioned, when we think about these very complex patterning or profiling using machine learning, this is certainly something we can do and do quite often. The challenge there is how we think about clinical implementation, and that oftentimes becomes quite a bottleneck. I think the value in our case is the fact that we can do both of these allows us to assess how much information can be gathered from one approach versus another.
All right, and I think we might have time to squeeze in one more question. In the last case presented, this is for the mitochondrial dysfunction marker. The biomarker is almost at the same level in baseline pre-treatment among all patients. How does it help in distinguishing response to future treatment?
Yeah, so I think there may be two cases there that are being sort of integrated together. So in the case of the mitochondrial marker that we identified there, this is a mitochondrial marker that's present at baseline, not with therapy, that denotes the degree of mitochondrial dysfunction that's occurring in vivo. Now, in that case, it turns out that the degree of elevation in that marker is very much related to the therapeutic response to certain therapies. And what we see there is with institution of certain therapies, those mitochondrial markers begin to drop in individuals, indicating an improvement in their underlying mitochondrial biology or mitochondrial function. In the last case report that we showed there, this was in another rare disease not related to mitochondria. And what we find is, as suggested, the baseline levels of this marker are quite common in individuals. But after therapy, and this is a marker that's a pharmacodynamic marker, there's an increase in certain individuals, whereas other individuals have a very flat response. And what we find is that if you subset individuals according to that pharmacodynamic marker, you can begin to identify those that ultimately are responders versus non-responders. Now, clinical implementation of that is an interesting idea. And what you oftentimes find is that these pharmacodynamic markers can change very rapidly. In this case, it was 1 week after institution of therapy, whereas the mortality or the ultimate clinical outcome may be many weeks or many months or even many years down the line. So in this case, you can imagine an enrollment strategy may include enrolling patients on a therapy, measuring a pharmacodynamic marker, and only including those individuals that have a positive pharmacodynamic response for the duration of the clinical trial.
Well, thank you very much, Mo. It looks like that is all we have time for today. I know there are a couple of other questions that we didn't get to, so Mo and I will be sure to follow up with you directly after the session. Thank you again to everyone for attending, and Mo, I'll let you close us out with any final thoughts.
Thank you very much. Thank you, everyone. And again, Tanya, and to the AAPS, appreciate the opportunity to share some data with you today. And we'd be happy to follow up with anyone who's interested in these approaches.
Thank you so much, Mo. Thank you so much, Tanya. And for everyone attending, there will be a recording of this presentation up on the AAPS website by the end of the week. Again, thank you so much to Sapient for this wonderful presentation, and you may all disconnect from the webinar. Have a wonderful day.
Thank you.